So I believe this is where we left off last time. We were just going over some of the emerging diseases, right? Ones that have increased in incidence over the past 20 years. And they could be new or old. They could have changed. So one of the problems with hantavirus and Lyme disease is this movement, encroachment of people into animal habitats, and the animals are the ones that harbor these particular diseases, and it's contact with them that transmits it to us. For Lyme disease, it's a bite of a tick, right, that has bit an animal that has the bacteria. What do you have to worry about for a hantavirus? What do you need to stay away from? Anyone know? Huh? Fleas, yeah, fleas. Mice and their fleas. And then changes happen within the infectious agents that can make them more infectious to other animals and humans. And so we've definitely seen this with HRV and SARS. And there was definitely the fear that SARS would travel worldwide, that it could become a pandemic. So this increased travel that, you can go from one side of the world to the other in a day also increases the spread of disease, right? Our ability to travel so easily. And that was definitely the fear with SARS. The unfortunate thing with HIV is it's already gone worldwide, right? And it's everywhere. And then for the problem with vaccination, and that, and nowadays too, you're pretty much going to get to the point where you're not going to have a choice anymore. Right? You used to be able to claim religious reasons or other reasons to not get vaccinated. Nowadays in the public school system, it's moving towards you will actually have to have a doctor's note, and you have to have some type of allergic reaction or some type of medical reason as to not be vaccinated. You'll no longer have the option to choose religious beliefs or any other beliefs for that matter. And that's because it puts other people at risk when we don't vaccinate, right? And we've seen a resurgence in whooping cough, which is also known as pertussis, and measles because of lack of vaccination. And those of us that can get vaccinated and don't have any medical reason on why we can't, we protect our society in a whole, and this is referred to as herd immunity, right? Where most of us in the herd, most of us in the group, if most of us are vaccinated, we protect the ones that can't get vaccinated, right? We create this buffer zone for them, where if we don't get vaccinated and we get it, we become a source of infection for individuals who can't get vaccinated for medical reasons. So that's sometimes a sticky point for some people, right? And part of it is the misconception in the media and stuff, and the horrible belief that was spread around, this lie, that you could get, I'm so aggravated by it, I can't even remember. Autism. You cannot get autism from vaccination. It's been scientifically proven that you cannot get autism from vaccinating, okay? It's just sad, right? That sometimes the media and the spread of misinformation does not help us at all. So be very wise about what you read, right? And the source of where you read it from, right? And as my professor in grad school said, read widely. Don't just use just one source. Use multiple sources for information, right? Yeah, the answer was D, because none of these choices are correct, right? So which of the following reasons for current rise in emerging diseases is false? Not a single one of them was false. Every single one of them was true. And remember, when they ask you things like that, it's best if you take each statement as a true and false statement. Thanks for stopping me on that. So, pathogenic microbes, including bacteria, which are our main focus a lot of times, fungi, notes, not so much, protozoa, not so much because we have limited exposure to them. Viruses, though, are another big one for us. All of these can cause damage to the tissues of our body, lead to the symptoms of the disease, right? So when you have a rash, when you have fever, when you have nausea, when you have a headache, when you're vomiting, these are all things that could happen, and it's a response to what's going on in your body, right? These symptoms, right? These signs, what you're dealing with. These organisms or viruses, they're using our body as a habitat, a place to live, and for multiplication, right? They're eating us, or in the case of viruses, using our cells to make more copies of themselves. They'll persist, right? They'll stay as long as we don't eliminate them, or our immune system doesn't eliminate them. They'll gladly make you their permanent home, right? And they also use you as transmission to others, right? To give their babies new places to live, right? The disease symptoms are often also offshoots of our defense mechanisms. What this means is that when we have fever, sometimes it's the organisms that's inducing our body to increase in core body temperature. Other times it's our immune system responding that something is foreign and increasing our core body temperature in hopes of killing that organism, getting too hot for that organism to survive. Also, our body moves faster, chemical reactions happen faster when you're warmer. Right? So that's one of our defenses. A rash is usually our immune system attacking the cells that are infected. Right? So that's showing you that we're actually responding to what's there. This too can be damaging to us. Right? Inflammation, a major component of our immune response that we're going to talk about in the second module in this course, is white blood cells rushing to that area. Well, in that process of the white blood cells getting there, fluid also leaks out of our blood vessels into our tissues. This can create a serious problem in our brain and spinal cord because it's encased in bone. Bone is not flexible, right? It can't expand for this excess fluid in the area. So it just creates more back pressure on the brain and spinal cord, right? And can be quite life-threatening. Same thing with our lungs. Although we need a certain amount of moisture for gas exchange to happen, if you have too much fluid in your lungs, right, you can't have enough gas exchange happening. And so therefore breathing is no longer possible, right, and you can die. An infection of the lungs, right, pneumonia, just refers to an infection of your lungs. It can be caused by a bacteria, by a virus, even a fungus, right? Lots of different things can cause that infection. When we say meningitis, we're referring to inflammation of the meninges, which are the membranes that surround your brain and spinal cord. Again, it could be caused by a multitude of different things. It could be a bacteria. It could be a virus. It could be a fungus. Okay? Could even probably even be a protozoa. Right? When we say itis, we're just referring to inflammation. But that's actually a good thing sometimes, because inflammation means your immune system is responding. Right? The problem is when your immune system is responding and it keeps coming in and doesn't eliminate the problem. Right? When you have long-term inflammation, right? When your immune system isn't able to take care of the problem. And then the inflammation itself can be a problem. Which drug that you might have in your medicine cabinet impedes inflammation? Anyone know? What? Ibuprofen, or Motrin is the brand name, right? Or ibuprofen is the drug name. You should not take that unless a doctor tells you to, right? Because you impede inflammation when you take that. That's the main way your immune system works, is to get white blood cells to the area that's experiencing damage or invasion. If you take ibuprofen, you're impeding that ability of your body, that very important first step of your immune response. Now, of course, as I said, if it goes on too long, right, it can be damaging. So there are times to take ibuprofen, right, but usually I would not do that unless a doctor tells you to do so. Right? Tylenol is a different story, right? Acetaminophen, right? that is a fever reducer, it's a pain reliever, right? But also should not be taken on a regular basis, right? Should be taken as needed. If you're taking it every day, you might want to go see the doctor, figure out what's wrong, right? You might be hiding an underlying condition, right? And you're just making it worse. So we use these things sometimes without thinking about what we're doing. So hopefully this class will even improve our health, right? We might even become germaphobes. I hope not, right? Just wiser about the world around us as it relates to microscopic organisms. So as it relates to organisms, when we refer to them by their scientific name. In the binomial system, we use both the genus name as well as the species name when we're referring to the species. I always like to think of it as bacteria have first and last names just like we do, right? So two names for them. So in that naming system, which should hopefully be reviewed for you, the genus name is always capitalized. So in the case of Escherichia, you see that it's capitalized. The second word is the species name. It is always lowercase. You never capitalize it. So when I was in grad school at Southeastern, we had t-shirts made up, right, for the biology program. And their mascot is the lion. So we had put the classification scheme on the back of the shirt, you know, kingdom, phylum, class, right? And they got down to the species name, and unfortunately, they decided to capitalize the species name on the t-shirts. We went to go pick them up. We're like, oh my God. And they're like, we're not reprinting them. It's not our mistake. It's your mistake. I'm like, I seriously doubt the person who submitted this order put a capital letter as species, right? It's a graduate level biology student who did that. They would not have made that mistake. So like I like to say in my biology lab here, microbiology lab, we use it as a teaching moment, right? We're like, what's wrong on our t-shirts, right? Unfortunately, what is wrong on our t-shirts, right? They had capitalized. A lot of times, especially in microbiology, we abbreviate the genus name. And so a lot of people, even to that point, refer to Escherichia coli as its abbreviation, right? We always say E. coli. I wonder how many people actually know the genus name because we abbreviate it so often. So the other thing that you'll run into with bacteria is some of them are quite different from other species within the group, but not different enough to get a whole nother species designation. So we have it's referred to as subspecies, but more oftenly referred to as strains, right? So E. coli or Escherichia coli 0157H7 is a particular strain, right? So the strain designation always comes after the species. This particular strain acquired a very mean toxin from Shigella. It's called the Shiga toxin. And this is the one that causes what is referred to now as Shiga toxin E. coli infections. And this is a very serious infection. This strain of E. coli you do not have in your gut. It's pathogenic. If you had it, you'd be in the bathroom or in the hospital. Okay? Causes really serious destruction to your intestines. So this is the one that when we have contaminations of our food supply, way back when, they pulled all the spinach off the shelves at one point, and how the spinach got contaminated was fecal runoff from a nearby dairy farm got onto the crops, onto the spinach crop, and that's how the spinach got contaminated. So it was actually, it's warm-blooded mammals that carry E. coli, right? But our crops, even like spinach, can get contaminated, right, by sewage contaminating the crops. So the CDC tracks this, right? Every once in a while we'll have an outbreak related to some food source that unfortunately is getting contaminated one way or another. So there's one other thing that you'll notice about the way these species and genus names are written. What do you notice? they're italicized, right? And I don't know about you, but I cannot, when I'm writing, italicize. So when you're handwriting something, what do you need to do? You're supposed to underline it, right? When you're writing a proper name, you're supposed to underline it. So here, which ones would be correct? Just one of them is correct. C only has one name, right? Just has coli. B, Escherichia coli, right? And it's italicized. The genus name is capitalized. The species name is not. If this one was an abbreviation, if it was a capital letter E dot coli, then it would be correct. Because you can abbreviate the genus name. But you got to have both when you're talking about a species. You got to have both the genus name and the species name. So the next thing that we're going to talk about is the different cell types that exist. And these might be review for you. All right, we talk about prokaryotes versus eukaryotic cells. Sometimes you'll see it spelled with a C instead of a K. All right, that is correct. It's an alternate spelling. I prefer the K, even though in my name it's a C instead of a K for Erica. And yet they always like to write a K. So pro means before, right? And so in this case, they believe maybe it was an organism before the advent of the nucleus. The nucleus, when you look at a cell under the microscope, a eukaryotic cell which contains a nucleus, it almost looks like a kernel, right? And so that's where karyote comes from. It actually means kernel, right? So before the kernel for prokaryotes. So prokaryotes, by definition, they lack a membrane-delineated nucleus. And therefore also, since a nucleus is actually considered an organelle, because it's a membrane-bound organization within a cell, prokaryotes don't have any organelles, any intercellular organelles. They don't have membrane compartmentalized sections that are specialized within their cells. Eukaryotes, on the other hand, U means true, are defined by the fact that they do have this kernel, this membrane-bound nucleus. And in fact, your nucleus is two sets of membranes that come together that form what we refer to as an envelope. And then, of course, they tend to have additional, right? This organizational system is present in eukaryotes. So they have things like mitochondria and chloroplasts and Golgi apparatus and lysosomes. They use membranes to compartmentalize different functions within their cells. Where a eukaryotic cell will have a plasma membrane, and it sometimes will also have membranes within the cell, but not organized to the organelle level. So when we organize life, right, you've probably learned in the past classification scheme, right, of kingdoms and phylums and all that good stuff. The good news is we're not doing that in this class, but there's one classification that may be relatively new to you that actually falls above kingdoms, and that's what we refer to as domains. So there are 3 domains of life. That's bacteria, archaea, and eukaryotes, or eukarya, which, as the name implies, makes up eukaryotes. So we'll talk about that cell type as it relates to each of these domains, how the cells are organized for those organisms that fall into those domains. We're going to look at a defining characteristic for one of those domains, and that's cell walls that contain peptidoglycan. whether they have membrane organelles or not, and just knowing cell type right there should tell you whether they do or not. And where would you find them on Earth, right? What type of environments do these organisms live in? Say that again? Moist, oh, yeah. Yeah, definitely like moisture. All living organisms actually need water. Remember, we mentioned viruses when we say the word microbes. Most microorganisms are included in that, and also viruses, okay, which most scientists don't believe to be living. So, again, this classification is just for living, so it excludes viruses. All living organisms are at least one cell. That one cell has the ability to reproduce, has the ability to have metabolism, right, chemical reactions within the cell. It can respond to its environment. It has a cellular membrane that defines it as a cell compared to the external environment. And it has genetic information. So, what's inside of a nucleus for a eukaryotic cell? What molecular substance we commonly abbreviate? DNA. What does DNA stand for? Yeah, deoxyribonucleic acid. Okay, that's in the nucleus of a eukaryotic cell. That's our genetic information, right? Stored in that molecular molecule. Do prokaryotes have DNA? They do. They have to. That's their genetic information. You got to have genetic information. The difference for them is it's just not encased in a membrane. Right? That's it. It's still there. So all living things, right? All living cells. Carl Woese came up with this classification in 1977, and it wasn't until probably the past 10 years or so it's made it into your guys' textbooks that it's been widely accepted in the scientific community. So when I was a kid, in learning the classification scheme of kingdoms, phylums, there was a little saying that I always remembered was, "Kevin, please close our front gate slowly." Right, so that was kingdoms, phylums, class, order, front, families, right, and then genus, and then species. So I gotta change my sentence a little bit because I gotta add domains above kingdoms now, right? So I say, "Damn, Kevin, close our front gate slowly," right? For D for domain, right? So domains, remember, are above all of the rest of that. Okay? So for living organisms, we have bacteria, archaea, and eukarya. And as the name implies, eukarya, are they prokaryotes or eukaryotes? Eukaryotes. And bacteria and archaea are both prokaryotic. And at one time, bacteria and archaea were kind of lumped together, because they were very similar, right? They're the same cell type to start. And the other unique thing about prokaryotes is they are single-celled organisms. They're all unicellular. So a bacteria is a single cell. An archaea is a single prokaryotic cell. And a lot of times people use prokaryote and bacteria interchangeably, but you're going to remember, right, we do have archaea, which are distinctly different from bacteria when we talk about prokaryotes. Although most of the information we know about prokaryotic cells comes from the study of bacteria. So as the name implied, eukarya, eukaryotic. Now we're eukaryotic, right? Our cells have a nucleus. Protozoa, right, which are single-cell organisms that are eukaryotic. So for eukaryotes, we can have both, right? We can have unicellular organisms that fall into this domain, and you can have multicellular like ourselves. Of course, this is a microbiology class, right? So other than talking about the humans, which are related to humans and pathogens, right? We'll mostly focus on unicellular, right, which would be microscopic microorganisms that are eukaryotic. So they have a nucleus. Cell walls containing peptidoglycan. Only one of these domains have that feature. Anyone know which one? It's bacteria, right? Bacteria are the only ones that have peptidoglycan. That doesn't mean that archaea don't have cell walls. They just don't have cell walls that contain peptidoglycan. And some eukarya even have cell walls, right? But our stipulation here is peptidoglycan. And that is a unique chemical structure of the cell walls of just bacteria. And that's important when we talked about penicillin last time, right? And we talked about how penicillin affects bacteria. This is why it affects bacteria and not our cells, is because it actually targets that cell's ability to make peptidoglycan, to make a structurally sound cell wall. That's how it damages those cells. Okay? So we're being selective in that aspect, and so is the fungus that makes it, right? That they're attacking bacteria because they are the only ones that have peptidoglycan. And that's what penicillin interferes with. So membrane-bound organelles. What cell type is the only cell type that has that? Eukaryotes. So no for our prokaryotes, right? Only yes for our eukaryotes. Where could you find bacteria? She said moist, right? And any living organisms likes a nice moist environment because you need water. Everything needs water. Every living organism needs water. So where on this earth? Could I go to Antarctica? Could I go to the ocean? Could I go in our bodies? Could I go in the soil, in the air? Well, you name any place on earth, you could find bacteria. So really all environments. In general, bacteria can be found everywhere. All environments imaginable on earth, we've found bacteria. Eukaryotes, I was going to skip archaea. Archaea, which are also prokaryotes, these guys are a little bit of a specialist. We didn't start finding these guys until we really started looking at very unique environments. What we would consider extreme. Highly salty, very hot, very cold. But you could find them just about anywhere. But these guys tend to prefer the extreme environments. In contrast, eukaryotes, how do we feel about extremes? Very salty would kill us, right? Very cold, Antarctica, I don't see any humans living there year-round. No. Yeah, they have found bacteria there in archaea in those extremely cold environments. Hydrothermal vents in the ocean's floor, nearly boiling water, they found microorganisms. Okay. You won't find us there though, right? So eukaryotes, right, we pretty much like anything that's not extreme. Right? You're not going to go to the extremes. You're not going to find humans living on Antarctica. And in fact, the penguins that go there, right, they're not even really they're not living there, right? They just hang out. They live in the ocean. Okay. They're just using it as a resting spot. You're not going to find us living, right, in the hydrothermal vents in the ocean. But you'll find bacteria there in archaea. So then when we look deeper into eukaryotes, we can do a little bit more of a grouping. And these are kind of unofficial groups, right? So they're not necessarily kingdoms or anything like that. And mostly we're looking at algae, right? And fungi and protozoa. Sometimes protozoa and algae are lumped together in a classification referred to as protists. So for cell organization, algae can be unicellular or multicellular. And of course, in a microbiology course, you would concentrate on the unicellular algae. Fungi as well can be unicellular, and typically our unicellular fungi are what we refer to as yeast. Your multicellular fungi that are microscopic are molds. And most of us are familiar with mold, right? Unfortunately. And then anyone know anything that's really macroscopic, multicellular, like you don't even need a microscope to see it, that would fall under fungi? Some of us like to eat them even. Mushrooms, right? They fall under fungi as well. Protozoa, these guys are the only ones that are just unicellular. Algae have cell walls. A lot of them are similar to plants in that they contain cellulose. Fungi, they'll have cell walls as well, some of them. Theirs tends to be made of a different substance referred to as chitin. Protozoa do not have cell walls. Can anyone think of an example of a protozoa you may have looked at in biology class that you'll find? Well, let's say, what type of environment would you find protozoa in, do you think? What sample would you be looking at in biology lab and see these guys swimming around? Swimming around. Aquatic environments, right? The ocean, lakes, ponds, right? And aquatic type environments. Paramecium, right? Amoeba, pathogenic things like Giardia, right? These could be found in contaminated water supplies. So for algae, since I said they're similar to plants, how do you think they get their energy? From the sun, right? They photosynthesize, right? They have the specialized organelle we refer to as chloroplast, right? That allows them to do that. They use sunlight. Fungi, on the other hand, ingest organic compounds. They can't make their own energy, right? They eat other organisms, whether living or once living, right? If it's living, they're considered a parasite. If it's not living, they're a saprophyte, which most fungi are saprophytes. So when things die, right, they are going to eat it, and then that helps add it back into the cycle of life. Most of us are saprophytes, right? We eat stuff that's dead. Organic matter, right, that was once living. I don't eat the cow while it's still alive. Prefer the no-mooing kind. Right. Plants, that's a tough one, right? Is it still alive or is it not? Right? They don't necessarily have to be in the soil to still survive, some plants, but for the most part, I guess you could say they're dead for those of us. Right? When we eat our plants, our veggies, our fruits. Protozoa as well need to obtain organic compounds, so they're eating stuff that's living or once was living as well. So in the case of an amoeba, it'll eat a paramecium, right? Paramecium will eat bacteria and yeasts and things like that. So where on Earth what type of environments would you find algae? You're mostly going to find those in aquatic environments. For fungi, where are you going to find those? On land, right? What we refer to as terrestrial. Mostly. Some of them are aquatic, but mostly they're terrestrial. You're going to find them on land. Protozoa, you can find in both environments. You can find them in aquatic environments. They're more prominent, but they're also quite popular in terrestrial environments as well on land. Then that brings us to the non-living entities. Viruses, and then probably other stuff you haven't heard of before, like Viroids, and Prions or Prions, or what I like to call infectious proteins. So notice that I said the division of life, right? These are our domains for living organisms. Both prokaryotes are bacteria and archaea. When we look at eukaryotes, right, we've talked about algae and protozoan. Like I said, sometimes they're lumped together and referred to as protists. We have our fungi. And then also in the microbial world, a lot of times multicellular parasites, helmets, worms, right, are sometimes discussed. They're so infrequently discussed in this course that there are a whole supplement chapter that's online, right? So when you guys click the online resources and library for our book, you'll find a chapter on worms there if you're interested in those kind of things. Usually they fall into the subdiscipline as parasitology, right? And parts of the life cycle of these worms, although some of them are quite large, sometimes are quite microscopic, right? So they definitely fall into the microbial world as we learn about them, right? Some of them are so small you do need a microscope to see them, where others are quite large, just like with fungi, right? Anybody remember the name of a subdiscipline that studies just fungi? It's not muscle. It's mycology, right? Mycology. And then off to the side we have our non-living, right? Infectious agents that fall into the microbial world. So as I said, non-living, most scientists don't believe that they're living. They're commonly referred to as agents. Again, not organisms. They usually consist of a few molecules you would find in living cells, but they're not organized to the cellular level. They're obligate intercellular parasites. They actually have to get inside of a cell in order to replicate, because they use the host cell's machinery in order to do that. Outside of the host, they're referred to as being inactive. Now, a lot of times we use the term to kill viruses, right? How do you kill something that was never living to begin with? You really kind of can't, right? But what are we worried about? We're worried about them infecting us. Can we damage them in such a way that they become non-infectious? We absolutely can. We can damage them in such a way. For the general public, killing makes sense, right? It means that it won't hurt you anymore, okay? So think about it this way, right? It is an entity. It needs to be intact in order to infect us. If we can destroy it in such a way that it can no longer attack us, then we could quote-unquote kill it. Or the proper term would be inactivate it, right? Completely destroy it. Make sense? So there are some chemicals that we'll learn about soon that have that ability, right? Some that you use at home. And when we talk about viruses in general, we'll talk about the different types and how these different things work. A little bit earlier than you said. Okay, so when we look at these for viruses, they contain nucleic acid. And it's going to be either DNA or RNA. Not both. It's one or the other. So a lot of time when we talk about viruses, scientists will say it's a DNA virus. It's an RNA virus. They'll even get specific as to whether it's single-stranded or double-stranded, because it can be either for that as well. Viroids are just RNA, and probably why you've never heard of them, or viroids, I was thinking of viceroys, we'll talk about those guys later, is because they don't infect us, right? So if it doesn't infect us, we don't care about it, right? But as in scientists in general, these do exist. And then there's no nucleic acid at all for Prions or Prions. For viruses, they're going to be DNA or an RNA, and then they're going to be surrounded by a protein coat. At the very least, that is what a virus is. Nucleic acid and a protein coat. But that's enough to get inside of a cell, right, and make that cell make a whole bunch more viruses. Viroids are just RNA, right? So if they're just RNA, they don't have to contain any protein, no protein. Prions or Prions are in fact just protein. So sometimes just referred to as infectious proteins. What can get infected by a virus? Well, we know we can, right? Can bacteria get infected by viruses? Can algae? Can fungi? Yeah. Yep, they have viruses too. All living organisms, right down to poor bacteria, even have viruses. No. The good thing with viruses, though, is they pretty much stick to particular host groups. So human viruses stick to humans, bacterial viruses stick to bacteria. And the reasoning for that is that each one of those organisms are quite different, right? And so the virus has to be different to infect those different types of cells. So bacterial viruses are quite unique in their ability to be able to infect just bacteria. And animal viruses pretty much just infect animals. Plant viruses stick to plants. Same thing is true for algae and whatnot. Viroids, why you haven't heard of them, is mainly they infect plants. Right? So they're a problem for plants. Where Prions or Prions infect animals and humans. But remember, we are animals, right? We just kind of put ourselves on this pedestal sometimes, right? And there is quite a distinction, but we are still an animal. So I'm trying to remember. Yeah, okay. So this is an old question. I don't usually pick questions like this in our question bank. But this, again, is a good summary of some of the stuff that we've gone over. So, which of the following are ways that prokaryotes differ from eukaryotes? So we'll say, and let's change the sentence, right? So let's say, prokaryotes usually are smaller than eukaryotes. Is that true? Yeah, because it said usually, not always. It said usually. Prokaryotes lack a nuclear envelope. True, because we know that eukaryotes do have that. Remember, I said that's the two membranes combined. Most have a cell... Okay, let me start over. Prokaryotes, most have a cell wall made of peptidoglycan. This is a tricky one, isn't it? It could be true, right? Because we, again, said most, and we didn't say all, because who are we excluding if we were to say all? If we're talking about prokaryotes, remember, we can't just think bacteria anymore, right? What's the other group we have to think of? Archaea. Archaea. And it's only bacteria that have peptidoglycan. Still with me? Right? So you got to make those important distinctions. Prokaryotes lack mitochondria and other membrane-bound organelles. True, right? That's one of the defining characteristics, is not having organelles, right? Not having that key organelle of a nucleus. Prokaryotes lack a cytoskeleton. We haven't even talked about that yet, right? They don't. They have one. But I'll tell you, when I sat in your guys' seats, I was told they don't. And the reasoning for that is at that time we did not have the technology or the ability to see that they have a cytoskeleton. Right? It's very rudimentary. It's not as complex as eukaryotes, but they do have a cytoskeleton. So all of these are true, except for this last one, right? So in the book at one time, they were all true, right? But now we do know that prokaryotes do have a cytoskeleton. Last one, then I'll let you guys go. So a friend comments to you that all bacteria should be killed to make the world safer. Which of the following would likely result if such a scenario is true? So what would happen if we killed all bacteria? Certain foods such as cheeses could not be made easily. This would be true, but some bacteria contribute to the making of cheeses, right? So your answer is A. So would humans have fewer fungal diseases? No, because we're being very specific about the type of disease there, right? Fungi diseases are different from bacterial diseases. No medications could be manufactured. False, right? Some, like antibiotics, are made by microorganisms, right? But some of them are fungi, some of them are bacteria, right? So this one, the correct answer is A. So humans would be free of disease? Definitely not, right? Because we know fungi, viruses, other things make us sick. Would plants be free of disease? No, we just learned that they got viroids even, right? And viruses. If you came in late, check in with me. Just give me a second to